learning about insulin cell signaling. Let's start with the pizza. What happens when you eat this beautiful pizza? Well, it goes through the body, goes through the esophagus and into the stomach, and eventually into the intestines. The intestines eventually break down the pizza and glucose is produced. Pizza is a carbohydrate in this case, so carbohydrates produce glucose. What's significant about this glucose? Well, we need to get it into this cell membrane over here to produce ATP for our body to use. How does this happen? Well, let's look at the pancreas. The pancreas right here produces insulin. This insulin goes inside the cell membrane via the intracellular receptors and eventually starts the phosphorylation cascade process. So, let's talk about the phosphorylation cascade. Insulin right here enters the receptor. This receptor then triggers this relay molecule right here to activate inactive protein kinase 1, which goes and activates active protein kinase 1. This then activates inactive protein kinase 2, which activates active protein kinase 2. Something to note, whenever an inactive protein kinase becomes active, ATP turns into ADP, as well as these active proteins have a phosphate attached to them. So this phosphate, what's going to happen to it is this protein phosphate, it's a catalyst, an enzyme. It removes this phosphate so that this process can repeat itself. Basically, this removes this phosphate to make this inactive again so this process can keep repeating itself. So let's continue on. This happens to all the proteins in this, so including these ones, and these ones. So, active protein kinase 2, what happens to it? It goes and activates this thing called the inactive protein, activates the active protein. Remember, ATP turns into ADP from inactive to active, and the phosphate attached to the active protein is removed by this protein phosphate enzyme so that this process can repeat itself and this will become inactive again. Like, the next ligand of insulin can start this process. So, this active protein right here triggers a cell response, and this GLUT4 goes all the way up to the membrane to absorb the glucose. That's how the glucose gets in. It just passes through these things called the GLUT4. It has this kind of little tube here, and just crosses through that. Now let's talk about amplification. Alright, so something I wanted to talk about. Amplification. Right now this diagram only has two active proteins. Amplification will have a secondary molecule such as cyclic or CA, and that will make it so that there's going to be more proteins. So it'll be active protein kinase 1, active protein kinase 2, maybe active protein kinase 3, 4, 5, so on, however many the cell needs. That is amplification in a nutshell. Alright, let's talk a little bit more about this glucose. So once the glucose enters the GLUT4, it is used for ATP, for glycogen storage, and triglycerides. That is all you need to know for insulin cell signaling. Thank you.